Now let's discuss the Starling curve. The concept behind the Starling curve will definitely be on your Step 1 exam in one way or another. Board examiners love this because it gives them a chance to test physiology using a diagram. Diagrams or figures that require a lot of conceptual understanding tend to appear in a lot of board questions. They usually show up on these exams as a visual. They usually do not have labeled axes either. Another great example of a diagram you might see on your Step 1 exam is the oxygen hemoglobin dissociation curve. So the Starling curve plots ventricular and diastolic volume, or preload, versus stroke volume, or cardiac output. Remember, we can write that because cardiac output is determined by heart rate times stroke volume. Starling really just wanted to see the effect of volume. So why did our buddy Frank make this curve? He wanted to show that stroke volume can be influenced directly by how much the ventricle is distended by a given blood volume in diastole, which is our preload. So the amount of distension of the myocardial wall directly reflects the amount of blood in the chamber, which is the end diastolic volume, also termed the preload. The tension produced by the chamber distension stretches the myocardial sarcomeres. This will allow more opportunities for interactions between myosin and actin in the subsequent power stroke. Remember that this is only beneficial up to a certain amount of stretch. You can actually outstretch the sarcomeres, causing a decreased ability to contract back to normal. Now, what would cause a downward shift in the Starling curve? How about heart failure? Of course, that's not what examiners are going to ask you, so let's understand why there is a shift of the curve down and to the right, which is something they might ask you. What is occurring in heart failure? So you can either have eccentric or concentric hypertrophy, but both eventually lead to some sort of systolic dysfunction or diastolic dysfunction. Now let's review quickly. Eccentric hypertrophy occurs mainly due to volume overload, while concentric hypertrophy occurs due to pressure overload. Now we just discussed concentric hypertrophy, so let's quickly run through that. So remember, someone has an increased afterload, which means that we'll have to really flex that left ventricle. So the left ventricle will increase its wall thickness because we add sarcomeres parallel. And this increases our wall thickness at the expense also of decreasing the radius because we are adding sarcomeres towards the center. So this is where the blood will be, and obviously there's less space available for blood to be when you're thickening the wall towards the middle. We added so many sarcomeres in parallel to help push against a very high afterload that we eventually made the ventricular wall incredibly stiff, and it cannot relax to allow adequate filling in diastole. Just like that huge guy at the gym that is so much muscle, he looks like he can never be loose or relaxed, and we've done the same thing to our left ventricle. The stroke volume will be low secondary to a very low end diastolic volume. There's not much space here in this chamber to put blood in. Now in the case of eccentric hypertrophy, we had a lot of volume, so we needed to stretch out our ventricle, while at the same time we also needed to add some sarcomeres, but this time we did them in series, which means that we did them end on end on end on end, and now this way they can stretch really far. And this allows us to allow the chamber to expand, because there was so much blood overlaid in that chamber that we had to add all those sarcomeres in series, which is just like attaching rubber bands end on end. Eventually though, the sarcomeres get so stretched out, and the ventricular chamber itself. So not only did we thicken the myocardial wall by adding mass to it, because we added those sarcomeres in series, but we eventually also overstretched it to the point that it won't contract like we needed to. The left ventricle stops ejecting blood sooner than we want, and we are left with a bunch of blood remaining in the ventricle. As you can see on your diagram, the congestive heart failure in the situation of systolic dysfunction shifts out the ventricular preload to the right, because we have super high preloads due to blood remaining in that chamber. So this is our systolic heart failure that is occurring at higher preloads, because at the start of the next cycle we have all that blood left in there that we could not pump out from the last cardiac side contraction. Now diastolic function puts us on a completely different part of the curve compared to systolic dysfunction. In the case of diastolic dysfunction, which is secondary to concentric hypertrophy, the left ventricle is so stiff that the pressure required to push blood into that left ventricle has to be very high. So what will blood do? It will back up into the more compliant left atrium and pulmonary vasculature instead of fill that left ventricle. This is occurring now at lower preloads, and this is our diastolic congestive heart failure. In this situation, we are on the more left segment of the CHF curve because the end diastolic volume is actually decreased. Due to the poor compliance of the myocardial wall, our cardiac output drops because of the lower preload. There is no problem with contracting in diastolic heart failure, but there is a problem with ejection because we never put enough blood in that chamber to begin with. So how do we get that to go back up? How does digoxin help us shift the curve up and to the left? Well, let's talk about digoxin. Remember that it competes with the extracellular binding site of potassium on the sodium-potassium ATPase. By decreasing the function of the pump, there is a decrease in extracellular sodium, which means that there will be less gradient from extracellular to intracellular with respect to sodium levels. Now, which antiporter will have less sodium to use from the extracellular space as a driving gradient for pumping calcium out of the myocyte? That is our sodium-calcium exchanger. Remember, it used sodium to pump calcium out, but we have abolished this gradient due to the digoxin. The result is that we have increased intracellular calcium, which will induce calcium release from the sarcoplasmic reticulum. As we've said before, more calcium in the cell, more available to bind troponin C and cause a conformational change in tropomyosin so that we can increase the number of actin and myosin binding sites, which increases our contractility, and that shifts us back up the curve. It is not quite understood how digitalis causes an increase in parasympathetic tone to the SA node, but you better remember that it decreases the overall heart rate. Now, how does exercise cause a shift to the left of the entire curve? When we exercise, we activate the sympathetic nervous system and decrease the outflow of the parasympathetic system. The release of catecholamines binds beta-1 receptors in the heart, causing increased intramyocyte calcium, also the increased opening of the funny channels in the cells of the SA node, which increases contractility and heart rate respectively. And remember, heart rate and stroke volume comprise our cardiac output, so overall we get an increase in cardiac output. The binding of catecholamines to alpha-1 receptors causes increased venous return to the heart due to that equation that we said venous return is due to the difference between venous pressure and right atrial pressure. This causes an increase in venous pressure, and therefore a bigger difference between the venous and right atrial pressures, causing more return to the right side of the heart. Do not forget here that binding of beta-2 receptors causes a lot of vasodilatation in muscles. This is also mediated by local metabolites that we will discuss in a second. Now, what about the inhibition of the parasympathetic nervous system? It prevents unnecessary blood flow to the renal and gut arterioles because we want our muscle, heart, and brain to have the increased oxygen supply, not kidneys and the splanchnic circulation. Now, what about the local metabolites? What's going to happen in the muscle itself? During stages of cellular respiration, we eventually use up all the oxygen and cannot continue to make very much ATP. We accumulate lactic acid, carbon dioxide, potassium, and precursors of ATP because we can't make ATP, AMP and ADP. So AMP and ADP are the precursors of ATP, and they accumulate because we cannot make ATP without oxygen. Remember, oxygen is the final electron acceptor or proton acceptor, depending on what you read in the books. They're both part of the equation. So the ones to focus on here, ATP and ADP, because they are broken down into adenosine, which has potent inhibitory effects on the ability of vascular smooth muscle cells to contract, so we inhibit vasoconstriction. We will discuss adenosine in a bit more detail when we hit the autoregulation section. Excellent. It's time for our first flash quiz. The blank is the most common artery to be occluded in the evolution of myocardial infarction. Your options are the circumflex, right coronary, the left anterior descending. This is kind of a tough question because we haven't discussed pathology quite yet, but I assume that you'll know the answer considering how we discussed the importance of the answer when we discussed the normal anatomy. It is the left anterior descending. Remember, the left anterior descending is a branch from the left main coronary artery. It supplies the most surface area on its own. It supplies the anterior 2/3 of the interventricular septum, and via its diagonal branches supplies the anterior surface of the left ventricle. That's a whole lot of surface area. So if you cause any occlusion of the left anterior descending, you have a lot of territory, and you cannot compensate with collaterals.